Good morning, ladies and gentlemen. It is with great honor and immense pleasure that are we gather here today on this auspicious occasion of National Science Day. We are here to celebrate the spirit of scientific inquiry and innovation that propels our nation forward. It is with great honor that it gives me immense pride to extend a warm welcome to our distinguished guests. It is indeed a rare privilege for our college to host a Nobel laureate, a beacon of intellectual prowess and scientific excellence. I extend my heartfelt gratitude to all the dignitaries present here today for gracing this occasion with their esteemed presence. Your participation elevates the significance of the event. Together, let us embark on this journey of exploration and discovery. Once again, a warm welcome to each and every one of you. May this National Science Day be a beacon of inspiration and enlightenment for all of us. Now, I would like to invite on stage Professor Gregg Semenza, a Nobel laureate in medicine and a professor of genetic medicine at Johns Hopkins. Sir, may I please welcome you on the dais? Good morning. I would like to invite Dr. Srivari Chandrasekhar, former Secretary of Department of Science and Technology in the Government of India. Now, I would like to request Dr. Srivari Chandrasekhar Garu to present an introduction of the National Science Day and its theme in 2024. Please, sir.
Good morning, young boys and girls, the faculty of the GITAM University, and students of this campus. Greetings to all of you and a very happy National Science Day 2024. On behalf of the management of GITAM University, a hearty welcome to our chief guest today, Professor Gregg Semenza. And Madam Semenza, thank you so much for being with us today. We are so proud that this campus is hosting a Nobel laureate for the first time since this campus is conceived. It's also very important to realize that today also happens to be the National Science Day. My young boys and girls who are sitting in the audience, who are not heard of National Science Day, a quick introduction to the National Science Day. Sir C.V. Raman, whose photo was shown at the beginning of this program, was the one who discovered Raman effect on this day in an institute in Calcutta, which is part of the Department of Science and Technology. Unfortunately, I was the secretary to the department, and this institute was part of our ministry. Those young boys and girls who get an opportunity to visit Calcutta, they should make a point to go to the Indian Association for Cultivation of Science, where the first Raman spectrophotometer was made by Sir C.V. Raman with his own hands. There was no equipment available those days in India. He had to make his own spectrophotometer. and discovered this Raman effect, which went on to win the Nobel Prize for India. Every year on this day, Government of India announces a special theme. And for the last several years, we have celebrated National Science Day. Sometimes it was a National Science Day with women as the face of Indian science, sometimes a science for children, sometimes which was for the globalization of science, sometimes on agriculture. This time, this year, the theme of this has been the indigenous technologies for a developed nation. We all know, under the leadership of Honorable Prime Minister, India aspires to be a developed nation. We always claim that we are a developing nation or a fast developing nation. But the dream and aspiration for all of us is India is going to be the top 3 economies in the world by 2047. And how we can use the indigenous technologies to use the Indian platform to make India the leader of the world. Today, we're happy that we have a Nobel laureate with us. And this National Science Day would be celebrated across the country by various schools and colleges. I'm so happy that young boys and girls are around. Hopefully, someday you visit some national institutes in the town, go around and get excited and become scientists and make India really a Viksit Bharat. Indigenous technologies refer to innovations, techniques, and practices that have been developed locally, often over generations, to address the specific challenges or meet the needs of commutes. While we embrace indigenous technologies, which also foster inclusivity and empowers the marginalized communities by recognizing and also make this country a great nation. It also reduces the dependency on the foreign imports and enhances the national resilience in the face of global uncertainties. We also practice Make in India, Startup India. I also encourage all the young boys and girls, at some point of time, we all become job creators than job seekers. By embracing the indigenous technologies, Viksit Bharat cannot only achieve economic prosperity, but also preserve the culture, heritage, foster social inclusion, and pave the way towards a more sustainable and equitable future. Today, we are very honored to have a Nobel laureate in medicine who is going to address us and enlighten us on the work he has been practicing at the Johns Hopkins. Today, we also have privilege of some of the young students here in the audience to have a privilege of interacting with him after the lecture and also have a photo with him, which I'm sure all of you will cherish. And who knows, one day, one of you or several of you win Nobel Prize and come back to GITAM and give a chief guest lecture here in the next 30 to 40 years to come. With these words, I also wish all of you again a very happy National Science Day. We are ready to have the lecture by Professor Gregg Semenza, who got a Nobel Prize in Physiology and Medicine in 2019. Professor Semenza is a Johns Hopkins University School of Medicine professor of pediatrics, radiation, oncology medicine, and biological chemistry. I'm happy there is a word chemistry, sir, in your designation. Dr. Semenza is also a vascular program director at the Institute for Cell Engineering. He shared the Nobel Prize in Physiology with Sir Peter John Ratfill at William Kiln Jr. Above all, Dr. Semenza is one of the world's leading experts on the molecular processes of oxygen regulation. We all know oxygen is essential for the planet. Unfortunately, when we consume oxygen, we emit carbon dioxide. So we need to get a Nobel Prize if we can do a genetic engineering for all of us, that we consume carbon dioxide and give out oxygen so that there won't be any carbon dioxide load on the planet. Hopefully someday it will happen, and that will get a Nobel Prize. Born in Flushing in the New York City borough of Queens, Semenza grew up north in Westchester County. The eldest of the 5 siblings, he attended, I don't know, the Sleepy Hollow High School, where his biology teacher, Rose Nelson, inspired him to study the sciences. I do hope some of your science teachers also inspire you to continue to do science. This teacher transmitted the wonder of biology and excitement of understanding the subject at a fundamental level, says Semenza. He was the fellow founder of the American College of Medical Genetics. Currently, he serves on the editorial board of numerous journals such as Cellular, Molecular, and Cancer Research. In 2012, Professor Gregg Semenza was appointed to the Institute of Medicine. Throughout his career, he has received a variety of awards, including those received due to his extensive research in pediatrics and biomedical science. I invite Professor Semenza to deliver his talk, and I hope after the claps, we will have the pin drop silence for the whole talk. Professor Semenza, over to you. Thank you. Down to see your slides.
Okay, how do I control them?
Actually, in the Zoom, they're connecting with the other campus, the medical campus. So
There we go. Okay. Great. Oh, thank you for the kind introduction and for the opportunity to speak here today about life with oxygen. And everyone knows it's very important that we all have oxygen to breathe. And it's the most important thing that we can't do without. And if you dispute that, please hold your breath and I'll get back to you later. But seriously, let's all breathe well. What I'd like to do is to tell you a little bit about the journey that I took during my career as a scientist, and how we understand how the body ensures that every one of the cells in your body, so you have 50 trillion cells in your body, and each one of those cells needs oxygen every minute of every day of your entire life. And the system that we discovered ensures that there's a correct balance between oxygen that you use in every cell to make energy and the ability of your body to deliver oxygen through your lungs, your blood vessels, and the blood that carries oxygen to all of your cells. And so this was the scene in Stockholm in October of 2019. Is there a pointer here? No. You can see on the right side of the stage is the royal family, and in the middle is the statue of Nobel, and on the left side are the laureates, including yours truly. And this is when I received the award from the King of Sweden. This is the Nobel Prize. And you see on the front side, thank you. So on the left, you see the bust of Alfred Nobel, and it has the year of his birth and the year of his death. And he died on December 10th in 1896. And so the Nobel Prize is awarded on December 10th every year in honor of his death. Now, December 10th in Stockholm is a very dark day. There's not much sunlight in Stockholm in December, but nevertheless, it was a very beautiful ceremony. And on the back of the prize, you can see these classic figures, and in Latin around the top is a quote from the Latin poet Virgil that translated into English is, let us improve life through science and art. And so for scientists, of course, we try to improve life by understanding life better and by understanding how our body works to be able to develop new medicines to fight diseases, as I'll show you during my talk. Okay. All right, so where did we start? We started by studying a hormone that's made in the kidney. It's a protein called erythropoietin, but it's much easier to say EPO. And EPO is made in the kidney and then gets into the blood and goes to the bone marrow where the red blood cells are made that carry oxygen. And so EPO stimulates the production of red blood cells. So if you go to a high mountain in the Himalayas, there's not so much oxygen to breathe. And your body will sense that the cells are not getting enough oxygen, and the kidney will make more EPO, and EPO will result in more red blood cells being made. So it's a beautiful system to ensure that enough oxygen is delivered to all of your cells. And what we wanted to try to understand was, how does the body sense if oxygen levels go down? And then how does it produce more EPO? So we looked at the EPO gene and we found a DNA sequence. So you know that the DNA is in every cell of your body and it contains all of the information needed to make every protein that makes up your body, including EPO. And what we found was that part of the DNA sequence was important for allowing EPO production to increase if there was not enough oxygen. So we found this sequence that we now call the hypoxia response element, because hypoxia means low oxygen. So this DNA sequence is important for the response to low oxygen. And then we found a protein that binds to that DNA sequence, and we called that protein hypoxia-inducible factor 1, or HIF1. So a factor is just a protein. Inducible means that it's made in response to a stimulus, and the stimulus in this case is hypoxia or low oxygen levels. And what we found was that this protein, hypoxia-inducible factor 1, which we call HIF1 for short, is actually composed of two different protein subunits that we called HIF1 alpha and HIF1 beta, because scientists like to use Greek letters to name things. So these two proteins bind together, and then they bind to this DNA sequence, and that turns on the EPO gene, allows more EPO to be produced. And what we did was to look at the amount of these proteins in cells that we grew in the laboratory. These are cells called HeLa cells. They're a cancer cell. They were the first cancer cells to be established, and that occurred at Johns Hopkins in Baltimore, where I work, in the 1950s. And these cells are used for research in labs all around the world. And what we did was we exposed these cells to different oxygen levels. So the oxygen that we're breathing now is the air contains 21% oxygen. So most of the gas in the air is nitrogen, but 21% of it is oxygen. And what we did is, as you can see, we exposed these cells to different levels of oxygen. And you can see that at the very low levels of oxygen, towards zero, you can see these curves go up very high. That means that a lot of these proteins are made when oxygen levels are low. And then as we increase the amount of oxygen that we gave the cells, you can see the amount of these proteins goes down very quickly. So we're able to show that these cells, in addition, in the laboratory, behave just like the kidney does in the body. They make more of the HIF protein, and that allows the cells to make new proteins in response to the low oxygen conditions. And so we wanted to know, how is it that the levels of this HIF-1 protein go down when oxygen levels go up? And what we found is that an oxygen atom is inserted into the protein, and that's a signal for the protein to be destroyed. So it's a beautiful system. If oxygen is available, the protein is destroyed. If oxygen is not available, the protein accumulates and can change the expression of thousands of genes in the body. And we first found EPO. This was the first gene that we knew was controlled by this system, but now we know of over 8,000 genes that are turned on and allow different cells in the body to respond to low oxygen levels. We also found that HIF-1 had other related proteins in the body that also responded to the low oxygen stimulus. So let's go back now to the kidney. I said that the kidney makes EPO, but there are many people who have what's called chronic kidney disease, where the kidney stops functioning. And as you know, the kidney gets rid of toxins that come from the metabolizing proteins. And of course, in the kidneys, the urine is made that goes to the bladder and then is excreted. So when the kidneys stop working, they also stop making EPO. And as a result, people who have chronic kidney disease become anemic, which means that they don't have enough red blood cells. And before 1986, they would require transfusions of blood. But in 1986, the EPO gene was cloned, and this allowed us to make EPO in the laboratory. And then individuals with chronic kidney disease could be injected with the EPO protein, and they would make more red blood cells. And this was a big advance for people with chronic kidney disease, because when you're anemic, you don't have much energy because you can't deliver enough oxygen to the body. The development of EPO as a drug was a big advance for the treatment of anemia in patients with chronic kidney disease. But still, this is an injection, and some patients did not respond to the EPO and remained anemic. So the discovery of the HIF system made us think, well, what if we stopped the HIFs from being destroyed? If we increased the levels of the HIFs, maybe we could make more EPO. And so a number of companies developed pills that increase HIF levels, they increase EPO production, and they increase the number of red blood cells. And for the chemists in the audience, this is the chemical structure of one of these compounds. And this was the first drug that was shown to be effective in treating anemia. And the clinical trials for this drug were carried out in Asia. And now 5 of these drugs have been approved in different countries for the treatment of anemia in patients with chronic kidney disease. So you can see that we started with a very basic discovery, and this led to the development of a drug for a very specific condition. Well, the second part of the story starts with this gentleman his name was Eugen von Hippel, and he was a professor at the University of Heidelberg in Germany in the early 1900s. And he was an eye doctor. And he found patients who had unusual findings in their eye. It turned out that they had tumors in the retina, which is the back of the eye where light is sensed. And these tumors were found sometimes in multiple family members, and it turned out that they were inherited, so from the parent to the child, due to a mutation in a gene. And so that was the first discovery in Heidelberg in Germany. Then a pathologist is someone who examines bodies after the patient has died to understand why they died. And this pathologist, his name was Lindau, and he examined some patients who had these eye tumors and found that they had tumors in other parts of their body, including the kidney. And so this constellation of the eye tumors and the kidney tumors became known as von Hippel-Lindau syndrome, named after these 2 doctors who had made the discovery. And it turns out that in this disease, so the gene that is mutated and no longer functions is now called the VHL gene, okay, for von Hippel-Lindau. And the protein that's made by this gene is critical. So remember, I told you that when oxygen is available, an oxygen atom gets inserted into the HIFs, and that causes them to be destroyed. Well, the way that they are destroyed is that this VHL protein binds to the HIF with the oxygen inserted into it. And it's the binding of this VHL protein that leads to the destruction of the HIF protein. So when there's no VHL, there's no way to destroy the HIFs. And you can see on the left that we have cells taken from one of these kidney tumors and grown in the laboratory, either under high oxygen conditions, 20% oxygen, or low oxygen conditions, 1%. And you can see that the HIF-1 alpha protein is present in both oxygen levels. Then we put a normal copy of the VHL gene into these cells, and you can see now that at the high oxygen levels, 20%, the HIF-1 alpha protein is now being destroyed again. So it's the fact that the HIF-1 alpha, and also you can see at the bottom, the HIF-2 alpha protein is not being destroyed that leads to the formation of the kidney cancer. Because the cells on the left that have the very high expression of the HIFs will form tumors if we inject them into mice, but the cells on the right where we've corrected the production of VHL, those cells will no longer form tumors. So this provided genetic evidence that the HIFs were very important, at least for the formation of these kidney tumors. And it turns out that pretty much all of the critical aspects of cancer development, the things that characterize cancer cells compared to normal cells, We know that there are genes that are controlled by the HIFs that are important for all of these aspects of cancer development. And so it was very interesting when a small biotech company in Dallas in the US developed this chemical compound that you can see on the left side of the slide. And this compound can bind to the HIF-2 alpha protein and block its activity. And it turns out that the HIF-2 alpha protein is particularly important for the kidney cancer. And so this drug was given to patients with kidney cancer, and then the growth of their tumors was studied. And you can see this graph with all these gray, black, and orange bars. And you can see the orange bars are all going down below the horizontal. And that means in those patients, the tumors got smaller while they were being treated with the drug. The black lines means that the tumor size did not change very much, and the gray means the tumors got larger. So you can see for most of the patients who were treated with the drug, the tumor either stayed the same size or got smaller. So this was a very remarkable finding because all of these patients had kidney cancer that had spread beyond the kidney. And once this cancer had spread beyond the kidney, there was basically no treatment. But now there is. And so this is the opposite because the first drug I told you about increases HIF levels to treat anemia. And here we're decreasing HIF levels in order to treat cancer. So we have examples of both sides. Where in the disease state, the normal regulation does not occur, and then we can correct it through the development of these drugs. So again, this is how we take a basic discovery, try to link it to what people have observed in different patients, and then develop treatments to make life better for these patients who now can live much longer because of the development of this drug. Huh? What happened? Can we go back? Can we go forward? Yeah. All right, here we go. Okay, so this is where I should have started, but this is where I'll end. This is my high school biology teacher. Her name was Rose Nelson. She was a tiny woman, but she had a giant impact on me and many of my schoolmates who went into science and medicine as a result of her inspiration, because she was such an amazing teacher and was able to show us how exciting science was and made us want to be a part of it. And I can tell you that the life of a scientist is tremendously fun because we are able to go into the lab and come up with an idea and test it. And it's all based on our own creativity, the ideas that we come up with. Nobody tells us what experiment to do. We come up with an idea and test it. And if we're very lucky, it will lead to a medicine that will make life better for people. We get to meet people all over the world, other scientists who share our interest in scientific discovery. And so I have friends all over the world, including in India now. And so it's tremendous fun. And it's a great profession. And so I encourage you all to think about science as a career. And science is a career for men. It's a career for women. It's a career for everyone. Thank you very much.
Thank you, Professor, for the wonderful talk. You have spoken about the great stories of the interaction of oxygen with the cells. And now we understand why we need oxygen. Thank you so much. Now, with a wonderful message from Professor Gregg on why one should choose science as a career and become a scientist. While the work he spoke about certainly has got recognition with the Nobel Prize, but the last slide that a high school teacher, how she inspired him to become a scientist and went on to become a Nobel laureate is laudable. I do believe that our young boys and girls sitting in the audience are inspired by that last slide that you are going to be a scientist. Scientist is not just a profession, but it's also a passion. It is highly rewarding. You will have friends across the world and you can do what you want. If you become an administrator in a bank, you will do what your manager tells. But if you become a scientist, you do what you like. I think that's the message, Professor. Now we open for question answer. Professor Gregg has been kind to take few questions. And I open this for discussion. Maybe the mics are with some colleagues around. So first raise the hand, stand up, mic will come to you, introduce yourself and ask the question. Question has to be shorter than the answer. So now we'll look for first young lot in the back benches. Yes. Please introduce yourself and then ask the question, yeah?
Good morning, sir. I'm Jyotika from BSc. Sir, I want to ask you what inspired you to choose this career in science?
So as I mentioned, I was very lucky to have a science teacher who was very inspirational, and also I had the opportunity when I was in high school to be in a research program. So I was able to go in the summer to a laboratory and see what research was like, and for me that was very exciting. I wasn't very good at it at the time, but I really liked it, and it really made me feel like this was something that I would like to do. And when I went to college, I was interested in genetics, in how different traits were inherited, and at first, it was more from a research point of view, maybe the studies with fruit flies or whatever. But while I was in college, a family that we knew had a child born with Down syndrome, which is an inherited condition. And I became interested in human genetics and decided to go to medical school as well as graduate school, and to take care of patients with different genetic disorders, as well as doing research in the lab. So for me, that I really enjoyed being able to both take care of patients, because there you have interactions that are very satisfying right away. The lab, things take longer to get to a satisfying point, but it's also very stimulating because you're always trying to figure out a puzzle. So the two of them, for me, made for a very happy career. And then the third part also, of course, is that I teach. So I teach medical students and graduate students about genetics. And so those three things make for a very happy career for me.
Thank you, sir.
Thank you for your question.
Hello, sir. My name is Shesai. May I ask a question? Is IQ important in science career? IQ.
IQ.
Yeah.
Well, I think that all of us make do with what we have. That's certainly been my approach. And no, I mean, I think that everyone has the opportunity to contribute to any area that interests them. And I don't really see any, maybe I'm not quite getting your question.
There's
Can you
a,
repeat the question, please?
is IQ important in scientific careers?
Is IQ important?
Yes, sir. In some fields like physics, where IQ is more dominant.
No, I don't think so. I mean, I think that everyone, you know, has the opportunity to contribute, and I don't think we need to do an IQ test to find out who should be doing genetics or any other type of work.
Okay.
Thank you.
Thank you, sir.
The next question. I think someone should come with the mic this side.
Good morning, sir. I'm Manognya. First of all, we are very thrilled to have you with us today. Thank you so much for coming. So my question is more about subject oriented. So HIF factors and their role in cancer metastasis or their progression. If you can elaborate a little more on that.
Yeah. So, you know, so in a cancer, the cells are growing very rapidly. And of course, all of those cells are using up oxygen. And in the normal parts of our body, we have blood vessels that are bringing, you know, red blood cells and oxygen to the tissue. But in the cancer, those blood vessels don't develop normally. And so they don't work very well. So the oxygen levels in areas of the tumor are very low, much lower than any other part of the body. And that turns on many genes through the HIFs. And some of those genes are important. So for example, if you have an area of the body that's not getting enough oxygen, you need to make new blood vessels. They need to invade into the tissue. But the cancer cells use those same genes to invade into the body. So normal responses are used by the cancer cells to spread throughout the body. So the more hypoxic the tumor, the greater the risk that the cancer will spread. And that's through the action of the HIFs. So we think it's very important to inhibit the HIFs in cancer. And so we've also been developing drugs like the one I showed you that inhibit the HIFs. And we've shown in animals, if we put tumors into animals, we can block the growth of the tumors. We can prevent them from metastasizing, for example, from the breast to the lungs. And if we combine these HIF inhibitors with other drugs that reactivate the immune system, because the cancer cells also avoid killing by the immune system, if we combine those two approaches, we can completely eliminate the cancer in the mice. And even we can inject more cancer cells into the mouse, no tumor will form. So we've cured the animal of cancer. And we can do that in about 50% of the animals. So we're very excited and hoping that we can get our compounds to the clinic like the drug that I showed you in the talk. Thank you for that great question.
There's a young girl. Tell your name, school, and the question, okay?
Hello, sir. Myself, Yagnashi. I have a doubt. I have a question. Do you face school name Genesis, sir? School name Genesis.
I'm sorry.
Sir, my question is, did you face any challenges in your career? Did you face any challenges in your career?
Do I have?
Did you find any challenges in your career?
Oh my goodness, yes. Yes, so when I was in graduate school, so as I told you, I went to both medical school and graduate school. And so I started in a laboratory. I worked in the laboratory for a year and I got nowhere. So I said, I went to the program director and I said, I think I need to go to a different lab because this isn't working out. So I went to another lab and worked for a year and found out that all the work that I had done was no good because there had been contamination of a sample. So I had to start all over again. So I was very depressed, and I was in medical school, and one of the students in my medical school class, her father was a psychiatrist. So I went to him and I said, I'm very depressed. And I told him why. And he said to me, well, Greg, it sounds like you have a good reason to be depressed. So I felt better after that. And I went back to the lab, started over again, and finished all of the work in the next year. So this is part of science, is that you have to have a frame of mind where obstacles are challenges that are to be overcome. And when I was in college and worked in a lab, my lab mentor used to say, search and research. So you have to have this idea that you're going to have a problem, you'll have setbacks, but you can overcome those problems. And that's part of the fun of doing research, is overcoming such challenges.
Thank you, sir.
Good morning, sir. My name is Rohan. I'm from Genesis International School. I have a question for you. That is, what would you suggest for young scientists and young science thinkers for getting their mind set on a discovery? Like, if you want to do a discovery, how do you brainstorm and how do you come about with that?
So as I mentioned, it was very important for me that I got to experience a research lab, when I was still in high school. And so I would encourage you to, reach out to colleges or universities to see what kind of summer programs are available, so you can get a sense of what it's like to be in a research lab. Because even though you do science experiments in school, it's not quite the same. So I think, having an early exposure to a research environment is a good thing.
Thank you, sir.
These have been excellent questions, really excellent questions.
Good morning, sir. Myself, Yogita. I'm from DPS Miapur. So my question is that, what are the interesting things that only a science student can experience in a science career, according to you?
Oh, that's a good question. I think that in order to do science well, you have to have a very systematic approach to things, right? Because you're trying to figure out, do I go this way or that way? And you don't want to go too far down the road and find out it's a dead end, that there's nothing there. So you have to take a very systematic approach to problem solving. And that same approach is used, for example, by doctors when they make a diagnosis in the clinic. So it's a very useful training that can be applied to other problems in life that we all face. So I think scientists have some advantage there.
Thank you, sir.
You're welcome.
I think while we take the question here, I wanted to check with the organizers whether we could take questions from off campuses, or if they want to type their question on the media they are looking, we can read the question. Is it possible? They can speak also. So any offliners who are watching us want to ask a question, maybe raise your hand, and our computer section, our team will be able to turn the mic on for you to ask the question. Next question, please, in the auditorium.
Hello, sir. My name is Nikhil. It's a great honor to meet you, sir. So my question is, what were your feelings or what were your thoughts on receiving the Nobel Prize?
Yes. So, in the US, when they call, it's the middle of the night. So the phone rings in the middle of the night, and the first time I was too slow to get to the phone, it stopped ringing. So I said, well, if it's important, they'll call back. So I went back to sleep and the phone rang again. So this time I was much faster getting to the phone. And then after that, I was quite speechless. And of course, my wife was there also, and we were in bed, and she could hear what was being said, and she was quite speechless. So that was my response, was that I did not have much of a response at all. I was so surprised. And I'm sure they were wondering, how will this guy give a lecture? He doesn't speak. But it turned out okay. Yeah. So my reaction was surprise.
Thank you, sir. Congratulations.
Thank you.
Sir, I'm Hamsika from Pallavi International School, sir. I want to know, what is your first discovery as a scientist? Your first discovery as a scientist.
My first discovery of it as a scientist. Oh, that's an interesting question. So when I was working on my PhD, I was studying a family that had a different type of red blood cell problem called thalassemia, where the red blood cells are not stable and break down. And this is usually inherited due to a mutation in one of the proteins that make up hemoglobin, which is the protein in red blood cells that carries oxygen. And in the family that I was studying, it turned out that they didn't have a mutation in that gene, that it was some mystery gene somewhere else that was causing their disease. So in a way, my first discovery was a non-discovery.
Thank you, sir. May I ask another question?
Certainly.
What is your toughest discovery in your life? Toughest discovery. I mean, it was really hard to discover something in your life.
Well, this is the fun of research, is that, we think we're following a path, but we never know where it leads. And this is what makes it so exciting. So when we started this work, we had a very simple question about production of EPO. We didn't think it would lead to the discovery about the production of 8,000 other proteins. So this is what we call serendipity, that there are unexpected discoveries, and that's what makes science so exciting. So it's actually, we have an idea, we have a hypothesis. This means we think it's this way. Then we do an experiment to test the hypothesis. And if we're lucky, the hypothesis is wrong, and the answer is more interesting than the one that we thought. And many people don't understand that. They think the goal is to confirm the hypothesis. No, the hypothesis is just a way to find out what is true. And what is true is often something that we didn't expect.
Thank you. But I think, Professor Semenza, we will try to take a question online. Are you ready to next? Ready? Please take the next question. Yeah. I think we just try to take just the technology is working or not.
Good morning, sir. Am I audible?
Yes, please go ahead.
Good morning, sir. I'm Dr. Anand from GITAM Institute in Vizag City. I would like to ask, sir, from the presentation, you mentioned about injection with epoprotein increases oxygen. And also, sir, mentioned about pills which being available. So could you please give us some clarity about the pills available, and also what are the dosages to be given? And is it for a few days, or is it just a single dose?
Yes. So I tried to make the story simple for the students. And the more complicated version is that the oxygen atom is inserted into the hips by a enzyme called a prolyl hydroxylase. And this enzyme uses oxygen and alpha-ketoglutarate as substrates. And one oxygen atom is inserted into HIF1 alpha. The other oxygen atom splits alpha-ketoglutarate into CO2 and succinate, making this an irreversible reaction. The prolyl hydroxylase inhibitors that turn on HIFs are all alpha-ketoglutarate analogs. So they bind to the alpha-ketoglutarate binding site in the enzyme and inactivate it by competitive inhibition. Depending on the degree of anemia, the patient may need to take a dose of the pill, say once a week or several times a week. In other words, they're very good. You can titrate the dose to affect the desired outcome in terms of how high you want to raise the hemoglobin. Okay? And of course, yes, if you stop giving the drug, the hemoglobin will go back down. Now, you may wonder if the patients have kidney failure, how does this help? But it turns out that in the fetus, EPO is made in the liver. And we think that the drugs can reactivate EPO production in the liver. So even patients who have had their kidneys removed can respond to these drugs, probably through EPO production in the liver.
Okay, thank you very much, sir.
You're welcome. Thank you.
Thank you. Now we take a physical question now on the central party, please.
Good morning, sir. Like we all know, we can't succeed in a one go in any science experiment. Like how to face the fear of failure while doing an experiment? Like what are your suggestions to face it?
Yeah, so, it's very, when an experiment doesn't work, it's very difficult because there's two possibilities. One is somehow the experiment just wasn't done properly, and that's why there was a negative result. But the other possibility is that, the hypothesis was wrong, and that's why it was a negative result. So negative results are much harder to interpret than positive results. And in the lab, I have to decide, well, did the student not just do the experiment properly, or did we, have the wrong idea? And so that takes a judgment call to make those decisions. And usually we'll try to do it several times, maybe in several different ways, so we can exclude that it's just some technical problem that's causing the experiment to not work. But as I said, oftentimes it's because our hypothesis was wrong. And then if we ask another question, we might be able to understand better what's actually happening. So this is, what we deal with all the time, is trying to interpret our negative results and positive results. And then to design the next experiment based on the results that we got with the first one.
We will have maximum 3 questions. So 2 from audience and 1 offline. Maybe there and 1 here. Yeah. Okay, first you go here, please.
Hello, sir. This is Priyanka from pharmacy. How do you know what to research on? And did you plan to research hypoxia from the beginning?
Well, that's an interesting question. So when I started, I was interested in studying development and why do certain genes get turned on in certain cells during development. I was interested in using what's called transgenic mice, where we can inject genes into a mouse and then look at the expression of that gene. So I was going to study a gene called the factor VIII gene that's involved in blood clotting. And I called up a scientist who had cloned the gene, and I asked, could you send me this factor VIII gene? He said, yes, but there's this other gene that you might consider called EPO. And so based on his suggestion, I read about EPO, and I was very interested because, as I mentioned, it's made in the liver before birth and then in the kidney after birth. So we first started by injecting different pieces of human EPO gene DNA into the mice, and we were able to get mice first with expression in the liver, and then if we put in a bigger piece, we got expression in the kidney and the liver. And then we became interested in the regulation by oxygen. And that turned out, of course, to be the most interesting part of all. So again, you can see that this was serendipity. I did not set out to study the EPO gene or to study oxygen, but that's where things ended up. And we've been able to study many different diseases because oxygen is so critical for every disease. So in addition to studying cancer and anemia, we've studied cardiovascular disease and the role of the HIFs in preventing cardiovascular disease and many other diseases. So, and again, this was just by chance that we found something that was so important and involved in so many different normal and disease processes. But again, as I said, that's the excitement of science, because you can't plan it out. And, you know, we have to write these grants to get money. And for these grants, we have to propose what we're going to do for 5 years. And if I knew what I was going to be doing in 5 years, I would be so bored, I wouldn't want to be a scientist. So I write the grant and then file it away, because I know in 5 years, it's not going to apply because we're going to be some other place that we didn't expect. And that's what I really enjoy about science.
Thank you, sir.
You're welcome.
Yeah, next year, yeah.
Hello, sir.
Yes.
I'm Snigdha from Epistemo School. I would like to ask, you mentioned how the VHL gene produces a protein that like destroys the HIF. How does the VHL gene produce that protein?
Yes, so you know that you have 2 copies of every gene, one from your mother and one from your father. And patients who have the VHL syndrome, one of the copies of the gene doesn't work, and the other does work. But in the kidney, the other gene, the one that does work, also becomes inactivated. And now there's no copy of the VHL gene that works. And that's why the tumors form in the kidney, because there's no functional VHL at all. But you have to have VHL in all of the other cells of the body, or you would not form at all. Because we know that mice, if we engineer mice so that both copies of the gene are inactivated, the mouse dies during development. It's never born, because having the normal levels of the HIFs is too important for all the formation of the normal body systems during development.
One question from online.
Good morning, sir. I'm Dr. Lalita. I'm a pediatrician. I'm head of the Department of Pediatrics in Jimsar, Visakhapatnam. So I would want to know how these pills or the injections would work, and at what stage in ALLs and neuroblastomas in children.
I'm sorry, it's hard for me to hear. Could you repeat it slowly?
So how do the pills and injections which are discovered would be useful at what stage in neuroblastomas in children and ALLs also in children?
Yes, so most of the tumors that we've studied have been in adults. The most common types of cancers in adults. We haven't studied pediatric tumors, but in principle, it should be no different in the sense that there's hypoxia within pediatric tumors as well. But what we've done in many cancer types is to look at the levels of the HIF-1 alpha protein in tumor sections by immunohistochemistry. And it's been possible to correlate high levels of the protein in the tumor biopsy with decreased patient survival. So that gives us an idea that in that particular tumor type, the HIFs are contributing to the disease progression. I'm not sure whether those types of studies have been done in the pediatric cancers that you're interested in. But that I think would be the first step toward trying to answer that question.
Okay, sir. Thank you so much.
Thank you, Greg.
Thank
I think
you.
one boy seriously is very anxious. So this is really last, okay? Really last.
Sir, myself Sukhjit Behra from GITAM School of Pharmacy. My question is, what are the main challenges that you faced in HIV pathway?
Challenges
faced in HIF pathway, sir.
Well, so our challenge is, for example, now our challenge is we've made the discoveries in the laboratory, and can we go from treating mice with cancer to treating patients with cancer? And of course, that's a big jump because many things that are effective in laboratory experiments are not effective in the clinic. And so that's our major challenge right now.
Thank you, Greg. Actually, I wanted to ask the last question, but someone is bargaining with me here that I should not ask. So maybe last from the right side. Yeah.
Hi, Dr. Semenza. It's great to meet you. I'm from Srinathi International School, and I'm an entering Stanford undergraduate. And I had a question.
Congratulations.
Thank you. I had a question about bioethics in the field of genomic medicine. So have you seen an importance being placed in this area, for example, in fields like gene editing? And do you think it's important?
Absolutely. Especially now that the techniques for gene sequencing are very powerful, and it's much more difficult for example, subjects to be anonymous because people can figure out based on their gene sequence who they might be. So there's that issue. And then, of course, with regards to the potential to treat genetic disease effectively, which we never had before, but now have. So, for example, the first treatment is a treatment for sickle cell anemia, where a change is made that allows the expression of one of the globin proteins, and it basically cures these individuals of sickle cell. Of course, this treatment costs over $1 million. So, the question is that ethical? Because only some people will be able to afford the treatment. So there are many issues with regards to bioethics. And so nowadays, that's recognized. And, for example, we have an entire bioethics institute at Johns Hopkins where people are dealing with all of these types of questions because we consider it to be very important.
Thank you
for your question,
Greg, for answering all the questions from the audience. Still many questions, but because of the time constraint.
Can I say one more thing?
Please.
To the students, what you have to understand is that I was once where you are now. And you can be someday where I am now. Thank you.
Great message, Greg. We wish some of the young students here will take science as their career and hopefully become as important as Gregg Semenza. I invite the Pro Vice Chancellor for felicitations to our chief guest today.
First faculty or faculty? Who is coordinating that? Would be stage for it?
I'll just one hygiene issue. I think we are going to have group photographs with the Nobel laureate. So I request all of you to maintain decorum and remain silent. I think Professor Reza will call out students in group wise from the schools. We have a lot of school children coming from about some 20 other schools. Professor Gregg has kindly agreed to be very patient with all of you. So I think Dr. Reza. First, I would like to invite our faculty members of the GITAM. Please be come in that dais. So we'll take the group photo. So I'm requesting all the faculty members of the GITAM, please come here very fast. So we'll take the group photo. Yeah.
Thank you, Mom.